Hello, my name is Jim Schnabel, and I will be discussing a series of biochemistry talks which are derived from the seventh edition of the Found Principles of Biochemistry text by Lehninger, authored by David Nelson and Michael Cox. I picked this book because it offers, I think, a good fundamental way of understanding biochemistry, and that is from a mechanistic standpoint, that the various pathways and reactions obey strict chemical principles of bioorganic reactions, and it would help not eliminate, but certainly reduce the amount of memorization that would be required for such a complicated course such as this. I will start with a very long but I think encompassing definition of biochemistry. And biochemistry's definition would be the study of how inanimate molecules that constitute living organisms interact to maintain and perpetuate life governed by physical and chemical laws which affect the non living universe. It describes in molecular terms the structures, mechanisms, and chemical processes shared by organisms and provides organizing principles. Uh, Biochemistry is a very complicated field. It's very broad, and I'm hoping with this and what I think will be another 121 talks that are organized around this text that we'll be able to derive a certain understanding of the field and perhaps use it in the whatever endeavors are necessary to master this particular subject. My own field is I do have a doctorate in this field, specifically in nucleic acid structure and sequencing and associated enzymology. That was my field at any rate. So we will go on and discuss some elements of this. Biochemistry, the molecules are variously complex, but they're derived from smaller molecules, at least from an evolutionary standpoint, that under constraints of time, pressure, and heat, we get more and more molecules that are larger and more complicated, and have over time devised a means to recreate them. And so when we look at properties of living organisms, we look at a high degree of chemical complexity and microscopic organization that over time the particular functions of pathways are confined to certain spatial areas within a cell, which is the organizing component of living things. And these cells differ according to the broad category to which they belong. There's also a system for extracting, transferring, using energy from the environment. The trend, of course, is to have greater and greater entropy or uncertainty. The living organisms defeat this purpose by creating order within their component by using energy to maintain that degree of steady state and relative imperviousness to the surrounding environment or adaptations thereof. Thirdly, we have defined functions for each of an organism's components and regulated interactions among them. So when you have a living organism, you can have a heart and lungs and livers, or if you have plants, you can have organelles and chloroplasts and things like that. And again, there are interactions between these using chemical signals which are evolved over time to help gain some sort of adaptivity to the surrounding environment, which leads us to the fourth property, which is mechanisms for sensing and responding to alterations in the surroundings. So the chemistry reflects the external environment or entities that perturbate the organism. So if the organism is hot, or if there is a wound or something like that, there are adaptations that occur chemically to make all sorts changes and to that. A capacity for precise self-replication, self-assembly is a critical function, that the genome is replicated and the offspring or the results of such replication give an organism that either has identical or in higher level organisms a recombination of various genomes, and this is a function of living things that they have a capacity to have progeny and maintain a certain fidelity with the original parentage. And then finally, a capacity to change over time, a gradual evolution, which has survival value, of course, and these are the 6 things that really distinguish a living organism from non-living organisms. So with that information, we can look at cellular functions. The cell is the foundational structural entity with living things, and it provides certain structures in common. So the outer aspect is the plasma membrane, which is comprised of lipids and proteins, and this employs a hydrophobic barrier, and there are specialized transport receptor loci which actually shift over the course of the cellular dimension. So the membrane is not a static item, but it is a very fluid entity in terms of the receptors and transport mechanisms being able to traverse the length or the breadth of the cell plasma membrane. And there are also membrane enzymes and transport substances that are used in concert with this very complicated barrier. Interior to the plasma membrane is the cytoplasm, and the cytoplasm has the aqueous component cytosol, and then there are particulate forms that can be fractionated with differential centrifugation. So you have mitochondria, which is the energy component in eukaryotes, perhaps derived from bacteria in an evolutionary set. Chloroplast found in plants has the energy deriving organelle there. Ribosomes, which are involved in protein synthesis, and proteasomes. These are common to both. The eukaryotes have a membrane-enclosed nucleus where the genetic material is. In prokaryotes and archaea, which we'll be discussing shortly, there is no membrane. The DNA is free-floating within the cytoplasm, and this is comprised of plasmids, which are a fruitful source of research potential. For the cell dimensions, there is generally plant cells are larger than animal cells. For the energy for the cells involved in energy generation and production, those cells generally are smaller, and this is done to maximize the amount of oxygen contact to diffuse into the organelle or the cell to help create energy. For larger cells, they are constrained by the diffusion needs of the cell. So there's a certain upper limit that can occur. When we talk about the 3 domains of life, there is a common ancestor to all of these, but you get eukarya, which are the eukaryotes having a membrane-enclosed area of genome, genetic material. We are more closely related to archaea than the bacteria. Archaea are organisms that prokaryotes, and they are derived from and inhabit ocean depths, areas of extreme heat and pressure. They can comprise anaerobic and aerobic forms. The third broad category are bacteria. Bacteria, they inhabit soil, surface waters. They're involved in living in organisms or decaying organisms, and these are all prokaryotes. So when we look at the energy sources, we're basically subdividing these by the energy source, whether it's chemical or light. So the light are phototropes, the chemical are chemotropes. And then once you establish where the energy source is from, the light or chemical, is a carbon source, carbon dioxide, or are they organic compounds? So if it's light, you're getting, that's a phototrope, and carbon source, carbon dioxide is a photoautotroph, and organic compounds are photoheterotrophs. And if you're using carbon dioxide as a photoautotrope as your carbon source, is water being used to reduce that carbon dioxide? So if you answer yes to all three, then you get the category of plants that we commonly see: plants, algae, cyanobacteria. They use light, carbon dioxide, and water to reduce the carbon dioxide. If water is not used, these are anoxygenic photosynthetic bacteria, green and purple bacteria. And they would use carbon dioxide but not water. If they use organic compounds as a carbon source, these are photoheterotrophs, and those are your green non-sulfur bacteria. So that's the plant kingdom in terms of energy source and carbon source and what the final reducing agent is. The analogous situation exists in the animal kingdom and in certain bacteria. So if the energy sources are chemicals, these are chemotrophs as opposed to phototropes. So chemotropes use the chemicals. If the carbon source is carbon dioxide, these are chemoautotrophs. If they are organic compounds, they are chemoheterotrophs. And if they're chemoheterotrophs using organic compounds as a carbon source, is a final electron acceptor oxygen or not? If it is oxygen, these are all animals, most fungi, protists, and bacteria. If the final electron acceptor is not oxygen, if it's something else, if they are organic compounds, those are fermentative bacteria. If they are inorganic compounds as the final electron acceptor, those are some species of Pseudomonas. On the other hand, if the carbon source is carbon dioxide, those are chemoautotrophs, and those are the category of oxygen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. So again, the organizing principle for these domains of life are: the energy source is a chemical or light, carbon source is a carbon dioxide or organic compounds, and the final final electron acceptor in animals is oxygen or something else, and is in the plants is oxygen reduced by water or not. So the common category of plants that we are familiar with use light, carbon dioxide, and water to reduce it. And the common category of all animals, they use carbon source of organic compounds, with the final electron acceptor being oxygen. And that's the breakdown in broad sense right there. So when we talk about bacteria and archaeal cell characteristics, going from external to the internal, we talk about the cell envelope, which is a plasma membrane, proteoglycan coat. Archaea has a different lipid composition. Then there are plasmids, which are again small circular DNA segments in the cytosol, free-floating, and prokaryotes. Eukaryotes have larger cells than in bacteria or archaea, and they contain organelles. And the organelles, which we'll be discussing the function in due time, mitochondria and the plasmic reticulum, Golgi apparatus, peroxisomes, and lysosomes. The cytoskeleton gives an internal structure and organization to the cytosol, and these change with cell function. So the process of cell death will cause a breakdown of the cytoskeleton. You can see this process under microscopic. And then finally, if you transport into the cell, that's endocytosis. And if you transport material out of the cell, that is exocytosis, and these are under tight regulatory control. So if you're looking at the structure in bacteria, there's a difference between gram-positive and gram-negative bacteria. Gram-negative bacteria, to start with those, an example of that is E. coli, which is heavily characterized. The outer aspect is a lipopolysaccharide coat on the outer surface attached to the outer membrane. Within that outer membrane are small pores that allow specialized small components to go in and out of the cell, no large macromolecules. Then there's a thin peptidoglycan inner layer. then a plasma membrane on the interior, and then cytoplasm. So in order in the gram-negative, you have a lipopolysaccharide, outer membrane, peptidoglycan, plasma membrane, cytoplasm. If you're looking at the gram-positive bacteria, you go from the outer layer, which is a glycoprotein with polysaccharides present dotting the surface of that cell. So you have the glycoproteins and peptidoglycan, which is generally a thicker amount than, or a more prominent amount than you would find in the gram-negative, and then plasma membrane. So for the gram-positive, it's glycoprotein with polysaccharides, peptidoglycan, plasma membrane, cytoplasm. For arachia, you start with glycoprotein, pseudopeptidoglycan, then plasma membrane, and then cytoplasm. So macromolecules that you get can be comprised of monomeric subunits. The subunits can be linked either by covalent bonds or by non-covalent bonds, such as hydrogen bonds, hydrophobic interactions, and van der Waals interactions. And these are polymers, and they have molecular weights of about 5,000. As I stated earlier, you can get differential subcellular fractionation of tissue. You can take, say, a liver, part of liver, and homogenize it, and then you can subject it to differential centrifugation. And by doing that, you can eliminate cellular debris with low-speed centrifugation. As you go further up, you can separate out from the precipitate whole cells, nuclei, cytoskeletons, plasma membranes, and then with higher speeds and length of centrifugation, you can separate out mitochondria, lysosomes, peroxisomes, and then at the very end you can get small fragments of endoplasmic reticulum, and then maybe the large macromolecules at the end. Eventually the cytosol, or the supernatant of this differential fractionation, can have soluble proteins, and this is one of the ways that we can get separation of substances and characterize the precipitate more thoroughly, or the cytoplasm, or rather the supernatant, as events warrant. When you're looking at the eukaryotic cell structure, it has many things in common between the eukaryote and the plant cell. There are some things that plant cells have that animal eukaryotic cells do not have. The plant cell has chloroplasts, starch granules, thylakoids, which are light-driven ATP synthesis, with the thick cell wall, vacuoles, which degrade and recycle macromolecules, plasmodesma, which provides pathway connections between adjacent plant cells, glyoxysome, which has enzymes that glyoxylate cycle, and cell wall. These are present in plants and not in eukaryotes. Eukaryotes have one thing peculiar to them, and those are lysosomes, which degrade intracellular debris. These are not present in plants. Other items of more lengthy amount of material is present in common. Those include the nuclear envelope, plasma membranes, mitochondria, rough endoplasmic reticulum, nucleolus, nuclear envelope, cytoskeleton, Golgi complex, peroxisomes, and these are smooth endoplasmic reticulum. These are all in common between plant cells and eukaryotes. So that takes care of the introduction part, and we'll be progressing further along, and we will be discussing some elements of chemical foundations as we proceed. So thank you very much for listening. Bye-bye.